webinar session. As usual, we will start in 5 minutes, so 3:05, so everyone has the chance to join. A little bit less than the half is already in this room, and let's wait just 5 more minutes and then talk to you then. All right. So thank you again for joining this webinar from the Permeability series with the topic Permeability Assessment of a High-Throughput Mucosal Platform. My name is Yannick, and I will be your host for this webinar today, and where we will dive into the world of mucus-related drug delivery challenges, discover the latest advancements in vitro platforms designed for assessing drug permeability through mucus. This research opens doors for designing more effective drugs, ultimately enhancing treatment outcomes. We are happy to have Cosmin as our presenter for the first time here at the Permeability series. Driven by a profound curiosity and a desire to contribute to the forefront of pharmaceutical sciences, Cosmin pursued his PhD in pharmaceutical and biomolecular sciences at the University of Torino in Italy. During this time, his research primarily focused on investigating the permeability of drugs across in vitro non-cell-based mucosal surfaces. After completion of his PhD, Cosman started a postdoc position where he contributed to the development of mucin nanoparticles as a drug delivery platform. Currently, Cosman is based in Berlin at the Freie Universität in Berlin, where he develops mucin-derived biopharmaceutical with antiviral properties. Before we start with the webinar, a few things regarding the organization. We've opened a Q&A window where you can type in your questions at any time, and we will answer them at the end of the presentation. And we'll also provide the slides and the link to the recorded webinar at the end of the presentation. Without further ado, I give the word to Cosman. Enjoy the presentation and see you at the end at the Q&A.
Quick check if you can see my screen.
Yeah, very good. I make the full screen now.
Super. So hello everyone, and before starting, let me thank Yannick for the invitation. And I have to say that I'm really excited to be here today to share with you what was my research of my PhD at the University of Turin in the group of Professor Sonia Visentin. So today I will talk about permeability, about mucus, and how we try to combine these two concepts together. In particular, I will start with a broad introduction where we'll see why permeability is important in the early phases of drug discovery. And then I will show you why we should care about mucus when measuring the permeability of drugs. And afterwards, I will show some results from our last paper where we try to develop a system that can be used to assess simultaneously the permeability of drugs and to take into account the effect of mucus. And of course, by the end, we will have a moment of discussion with Q&A. So let me start really from the basics, from the beginning, by showing you the drug discovery pipeline. So this because you have to know that in spite of the advances of the last decades, today bringing a new drug in the market is still a quite complicated task. Because you see, usually pharma companies have this library of compounds, tens of thousands of compounds, which they screen based on specific properties and at each step, the best candidates are selected. The problem with this process is that, as you can see, it's extremely time-consuming. For every new drug, on average, are necessary at least 10 years of research and development. And most importantly, it's really expensive. So every new drug on the market costs on average more than $2 billion. And one of the critical steps in this process is when drugs start to be administered to people. So when they go into clinical trials. And in this phase, usually 8 out of 10 compounds fail. So 8 out of 10 compounds will never reach the market. And drug candidates fail basically for 3 reasons. This being, first of all, the efficacy. The drug simply is not behaving as we would like. Also, the safety issues. It's a really important aspect because in this case, the efficacy might be good, but the adverse effects are also significant, and the overall risk-benefit profile of the drug is not optimal. And really important, drugs fail because of pharmacokinetic issues. In this case, the problems might be because the absorption of the drug is not optimal, or the distribution, the metabolism, or the elimination as well. And along the years, medicinal chemists and pharmaceutical scientists understood that designing drugs with a better pharmacokinetic profile can strongly increase the chances of having a successful drug candidate. But how do we design better drugs? So how we design drugs with a better pharmacokinetic profile? Well, actually, there are different strategies. One of these strategies could be, for example, to select those drugs which could have a good absorption profile. So why absorption? Well, absorption is a critical aspect for the effectiveness of a drug, because you have to know that unless the drug is intended for a topical administration, in all the other cases, the drug, we say that the drug is effective only because the drug have been absorbed. So only because the drug is bioavailable, so it's available to be effective. And this implies that the drug has to cross to biological barriers like cellular membrane. But measuring the absorption of a drug, it's not an easy task, because absorption is quite complicated, complex phenomenon. It's an interplay between different molecular properties related to drugs like solubility, lipophilicity, the acid-base profile of the drug, and most importantly, it's related to permeability, which is defined as sorry, as the ability of the drugs to diffuse through cellular membranes, through barriers. And in particular, permeability along the years have proved to be a really good predictor of the in vivo absorption. And because of that, along the years, the scientific community developed different models to investigate the permeability of drugs, and so to predict the in vivo absorption. And to make a long story short, we have many different models to study permeability, starting from the most simple, which are definitely the in vitro systems, non-cellular based, so without cells, but only artificial membranes. And we can go up to ex vivo models where you study the permeability through whole tissues. And selecting the best model, it's always a trade-off between the cost and complexity on one side, and on the other side, the reproducibility and high throughput capacity of your model. And usually, as I was saying, in the early phases of drug discovery, pharma companies have a high number of drug candidates, hundreds of drug candidates, which in principle they want to screen in a relatively short period of time and with a quite good reproducibility. And this is why usually the first model to be adopted are the in vitro models based on non-cellular artificial membrane. Like, for example, the PAMPA system, which is the parallel artificial membrane permeability assay, where you have a membrane made of phospholipids which mimics the cellular membrane. The problem with these systems is that they consider only the cellular membrane as a barrier, but they do not take into account another barrier that we have, which is the mucosal barrier. So the mucus. Mucus, for those non-familiar with it, it's this hydrogel that is covering all the wet surfaces of our body. You see, we have a layer of mucus on our eyes, in our mouth, in the whole gastrointestinal tract, actually, and in our airways, and even in our reproductive tract. And on this epithelia, mucus plays different roles. First of all, it is our first barrier against environmental threats like bacteria, viruses, or air pollutants. But also mucus ensures a constant hydration and lubrication of the underlying epithelia. For example, think at the simple movement of blinking your eyes. Without the layer of mucus, this movement would be quite painful for us because of the friction of the tissues. And of course, mucus, it's an important environment because, for example, it ensures the growing and the thriving of our intestinal flora. And why should we care about mucus in the first place? Well, in terms of administration routes, if we consider the administration routes, you see that drugs administered by the oral, by respiratory, or by rectal or vaginal route, in all these cases, in order to be absorbed, the drug needs to cross to the mucus layer. And if we look at the small molecules which are today on the market, you see that around 60% of them are formulated to be taken by mouth, so by oral. So we are talking about a huge number of compounds. But of course, you might say, okay, but where is your point? I mean, in the end, these are all marketed drugs, so they might have a relatively good absorption. That is totally true. The problem with mucus starts to be when, in specific cases, like, the so-called mucus-related disorders like cystic fibrosis or chronic obstructive pulmonary disease, where in these cases the mucus is not physiological anymore, but it's a pathological mucus. This means the mucus is more viscous, it's more acidic, it's chronically infected by bacteria. This means it's chronically present with anti-inflammatory compounds, and because of that, even the standard treatment with medications might be difficult. For example, this is a short list of some of the antibiotics which are used in the cystic fibrosis treatment. And for these compounds, it has been reported in the literature that their activity it's reduced or it's modified because of mucus. Simply, these drugs are not able to diffuse anymore through the mucus because the barrier properties of mucus are too strong. And so the drugs are not bioavailable anymore. And to sum up this part, we can say that today, mucus, despite playing a strong effect over the absorption of drug, today mucus is fully considered in the early phases of drug discovery. But this happens because we simply don't have in vitro mucus models that could be used by pharma companies when assessing this huge amount of compounds. For example, if you want to use the cystic fibrosis mucus, the best model would be to use the mucus itself, so the mucus from patients. But the problem with the cystic fibrosis mucus is that it's really not suitable for high throughput purposes because it's extremely heterogeneous in terms of composition and viscoelastic properties. And also it's quite complicated to handle it because of the biological risk. And so during my PhD, we try to address these challenges by trying to design a platform that could simultaneously take into account the effect of mucus and at the same time measure the permeability of drugs. And so this project started with a collaboration with bioengineers from Politecnico di Milano. They have developed an in vitro pathological mucus model that mimics with quite a good extent the viscoelastic properties of cystic fibrosis mucus and also the composition of CF mucus. For the viscoelastic property, to recreate the gel-like behavior of mucus, they used alginate, which is a natural polymer produced by Pseudomonas aeruginosa, which is a hallmark of the cystic fibrosis infection. And alginate, in the presence of calcium, can be cross-linked, and after cross-linking, it's forming a gel. While for the composition, to mimic the real mucus, they used porcine gastric mucin as a model of MUC5AC, which is one of the mucins present into the mucus. And what we've done with this mucus was to couple it to the state-of-the-art permeability system. So we started by using it with the parallel artificial membrane permeability assay, which, basically it's a 96 well plate where you have two compartments which are divided by a membrane made of phospholipids. And more recently, we also coupled the mucus with the PermeaPad system, which in principle, it's very similar to Pampa. The only big difference is the membrane. Instead of having a structured layer of phospholipids, we have vesicles of phospholipids, and this mimics to a higher extent the epithelium. And what we've done was simply to put a layer of mucus on top of the phospholipid membrane. And we like to say that in this way, we recreated a kind of mucosal surface where the drug has to diffuse at first through the mucus layer and then through the phospholipid membrane. And regarding the workflow, the whole experiment is quite simple, quite straightforward. On the first day, we pipette the mucus in the donor compartment of the permeability plate, which you like to use. And we let the mucus cross-link overnight. During this phase, the mucus becomes a hydrogel. And the second day, the plate is ready to be used for the permeability experiment. And so you can simply pipette your drug solution into the donor compartment and put the buffer solution into the acceptor compartment. And after coupling the two plates, you can start the experiment. And in our case, we designed the experiments at a single time point, so only one permeability measure after 5 hours to ensure a kind of high throughput setup. And for the quantification, yeah, here you can use the model, the method that best fits for you. In our case, we wanted to have a high sensitivity, and we used liquid chromatography coupled to mass spec. And in principle, the system, it's of course suitable for 96 well plates, but you could upscale it even to 384 well plates. And after setting up the system, we had to select our data set, so the drug that we want to test. And at the beginning, we started by testing mostly antibiotics, because our whole system is focused on cystic fibrosis, and antibiotics are the standard of care for the medications. And at the same time, though, we wanted to have a heterogeneous data set of compounds. So we wanted to maximize the chemical variability of our data set. And because of that, we increased the number of tested compounds up to around 40. And we investigated the chemical variability by looking at the distribution within the Lipinski's rule of 5 and the Weber rule. And to make a long story short, we've seen that our drugs are very well distributed within the molecular descriptors of these rules. And also another method that we used to investigate the chemical variability was to plot the drugs that our data set into the chemical space of the approved drugs. And again, as you can see, the red dots, which represent our data set, are very well distributed into the chemical space of the approved drugs. And after proving that our data set is different enough in terms of molecular properties, we had to measure our control values, so our reference values. And these are represented by the permeability of the compounds without mucus, so only through the phospholipid membrane. And here the idea was, let's try to see if we can predict the permeability of the compound based only on the molecular properties of the drug. And to do that, we divided our data set into two groups, into two categories: high and low permeable compounds based on the geometrical mean of the data set. And for each compound, we calculated several molecular descriptors related to lipophilicity, to solubility, size, and so on. And we correlated the permeability with each one of these descriptors to see which are the descriptors that best correlate with permeability. And we found that when measuring the permeability through the PermeaPad plate, so only through the phospholipid membrane, the size of the drug and the solubility play quite an important role, because these were the best descriptors correlating with permeability. And if we plot these descriptors into a three-dimensional region, you can clearly see that the high and the low permeability group are on different region of the chemical space. And in the next step, we finally had to see how the permeability of this compound, so the reference value, how the permeability is modified by mucus. So what is the effect of mucus on these compounds? And again, we measured the permeability through the mucosal surface, and we've seen that indeed mucus behaved like a dynamic barrier, because we've seen that most of our compounds had their permeability reduced by mucus and was statistically significant, but the effect was not the effect of a simple barrier, because some of the drugs you see were not sensitive to the barrier of mucus, because with or without mucus, the variation of permeability was not significant. And also for some of them, the permeability was even increased in the presence of the mucus. And again, the idea was, let's try to see if we can predict the effect of mucus only based on the molecular properties of the drug. So we started again to correlate the permeability measured with presence of mucus with the molecular descriptors that we calculated in the previous step. But this time, the correlation that we have seen previously were not so strong anymore. However, we were still able to define some trends. For example, we've seen that the charge of the molecule plays an important role over the interaction with mucus, because, for example, positively charged drugs are interacting more with mucus when compared with neutral or negatively charged drugs. And as I was saying, for some of the compounds, we've seen that their permeability was higher in the presence of mucus. And this was quite a weird phenomenon, because in the end, you have an additional barrier, but your drug is running faster. And this was counterintuitive in the beginning. But then we found that this phenomenon, it correlates really well with the pKa of the drug. So the more acid is the drug, the more will be the increase of the permeability. And the explanation that our hypothesis of this phenomenon is a complexation of the drug with calcium. The calcium that I remember, we use it to cross-link the alginate, so to form the gel. And some of these drugs, so the acidic drugs, can form complex with the calcium, and the overall hydrophilic profile of the complex, it's more favorable for the permeation. And since we are approaching the end of this presentation, I would like to give you like a real example of how we use this system. The last year we had a collaboration with a biotech in Italy, and they are developing a peptide which is really interesting in the cystic fibrosis context, because this peptide can increase the activity of the CFTR channel, which is at the core of the cystic fibrosis disease, but also it can improve the respiratory function and reduce the inflammation. So it's really a very promising therapeutic candidate. And this peptide is supposed to be administered by pulmonary inhalation. So it was really interesting to see how mucus, which I remember to you, it's in cystic fibrosis, it's more viscous, it's overproduced, and it's a more stronger barrier. how this mucus can impact the absorption of this peptide. So we tested the permeability of the peptide using our system, and we've seen indeed that mucus reduced the permeability of the peptide. But more interestingly, we've seen that uh, the permeability measured with cystic fibrosis sputum, so cystic fibrosis mucus, was very similar to the permeability that we obtained using our system. So we think that this kind of information might be really helpful in the early phases of drug discovery, because you might formulate better your compound, and you might um, define better your dose, taking into account the amount of drug that you lose because simply the drug it's not able to diffuse through mucus. And also recently, we show that in addition of this permeability application, you could use these mucus models also to investigate the antimicrobial response of bacteria. Because in collaboration with Bactrigel, which is the company that producing this type of mucus models, they have developed a more advanced, let me say, version of this mucus with a structured gradient of structured gradient and an oxygen gradient. And we asked if how bacteria grown in planktonic and within this three-dimensional scaffold, how do they respond to antimicrobial treatment? And we've seen that both Pseudomonas aeruginosa and Staphylococcus aureus, when they grow into the mucus, into this mucus model, they are more resistant to antibiotics. And these results are more close to the in vivo scenario. So again, what I want to say is that these type of models can be really useful defining when defining the pharmacokinetic properties of your compounds. And with this, I would like to thank all the people that were involved in this project, in particular to my previous supervisor, Sonia Vicentin. And again, I would like to thank Yannick and Fabio for the invitation, for allowing me to share with you my research. And last but not least, thank you very much for listening. Hopefully you are still there.
Yes, yes, we're here. We can hear you, and now everyone can see us as well. Thank you very much for this nice presentation. Very insightful, I would say. And we already got a few questions. If everyone else has questions, please type them in the chat, or you can write them directly to us, to our email address, if you need more support, or if you can't disclose anything. Feel free. I will put the email address in the chat later on. But let's start with the first question, Cosmin. It's from Dominic. Does that mean that drugs prone to negative food effects with concomitant calcium intake should be analyzed with care using the mucus assay?
I'm not sure I have understood your question, but if you are wondering if calcium can modify the permeability of compounds, so the absorption, the answer is yes, because as we proved for the acidic compounds, the complexation with calcium can increase their permeability. And we show this also by investigating the complex formation. So we show that we found by mass spec the mass of the complex. And in terms of formulation, you could speculate that drugs formulated as calcium complexes could eventually have a higher absorption because their hydrophilic lipophilic balance it's more favorable to diffuse through to the cellular membrane. Because in the end, the drug, which is negatively charged, is now forming the complex, so the charge is not available anymore, and the complex will be more lipophilic. And yeah, I'm not sure if this answered your question. You can also turn on your microphone if you want, I guess.
Oh no, this is not
Ah,
possible.
okay.
They can they can just hear us. Yeah.
Okay.
Um, but I think Dominic will ask again if this was not satisfying to him.
Okay.
Because the next question is from him as well. Is there any research already performed or planned to investigate how bile components and/or excipients affect permeability across mucus?
I am not aware of any research, but I'm pretty sure there must be something. There must be something. And in principle, you could also design kind of mm, bile bile components, like a mucus embedding the components of the bile by maintaining the same scaffold to have the structure of mucus, but simply changing the chemical composition of the mucus. But I don't know, honestly.
Okay. Maybe we have to clarify with Bactigel as
Yeah,
well, if they have looked into this
yeah,
again.
yeah.
And the last question from, sorry.
No, I think, and I'm pretty sure they are also developing an intestinal mucus model. So in that case, the composition has definitely to be different. But yeah, you should contact Bactrigen maybe.
Oh, they are looking into intestinal mucus. We
Yeah.
are all already in contact. Yes. And the last question from Dominic is, do we have any idea on how many drugs are in reality mucus permeation limited compared to epithelial membrane permeation limited?
In principle, all the drugs which are administered by pulmonary inhalation, and especially in a pathological context, so we are not talking about only about cystic fibrosis, but COPD, asthma, and so on. All these drugs are subjected to the mucus barrier. Because I didn't mention, but the barrier of mucus, it's really complex, because you have a dynamic barrier in the first place, because mucus is continuously produced and shedded out from this epithelia, but also you have a steric barrier. So you could also think at nanoformulation. um, the, 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 the bigger is your drug, your active pharmaceutical ingredient, the lower will be the permeation through mucus. And for the small molecules, you have a chemical barrier that can be subjected to the interaction with mucins in the first place, but not only, with lipids and with other proteins present in the mucus. And so this is for the respiratory administration. Again, the oral drugs might also be subjected to the mucus barrier, but on this regard, I'm not aware of many publications where they have investigated specifically the effect of mucus over the absorption in the gastrointestinal tract. Yeah.
Maybe this is an additional question. The application of the developed mucus model for cystic fibrosis is just for this case, or do we have normal mucosa as well? And this, I think, what we meant. Bactigel is right now developing different type of mucus. Right now we're in a phase to do the saliva for the buccal mucosa, or yeah, for the buccal mucosa, to investigate absorption there. But they are planning to do different normal mucus types as well.
Yeah, exactly. So one of the advantage of this type of in vitro mucus model is the modularity. What I mean is the mucus can be tailored, so specifically tuned to the different type of mucus that we have on our district. So this was a respiratory mucus model, pathologic in this case, but Of course, you could change the composition of the mucus, for example, by adding DNA, which is a component of mucus, or by adding phospholipids or any other type of protein that is present in the mucus. And so you can create a specific composition for the district you are interested to investigate. Yeah.
So if you are interested in a different application, different mucus, you can reach out to us and then we will find a solution for your application, of course. There's one more question. How does the drug solution excipients affect the mucus layer? Like we use PEG, DMF or DMSO as solubilizers or co-solvents. Do you have any experience with this?
Yeah, I mean, in our system, we used 5% DMSO to ensure that our drugs are properly solubilized, and so DMSO was used in all cases. When developing the mucus model, at Politecnico di Milano, they investigated the stability of mucus to DMSO and to buffer, and they checked for the increase of mass and the increase of volume, and the variation with DMSO was quite small. It was below 10%, if I'm right. So this it's not a critical component for the structure of the mucus, I would say.
Any more questions? In the meantime, I opened the presentation or shared the presentation with you, the PDF. I shared our standard mail address. If you have any further questions, we can forward your questions to Cosmin as well. If you want to exchange your research topic with him, it's not a problem.
I will share my email here in the chat.
Oh, perfect. Thank you. So if no other questions arises right now, I think we're done for today. I would like to do another introduction for the next webinar. It will take place in December. I have to look it up at December 6th. It's a Wednesday as well, same time, same day, with Elisa as well. She's presenting another research result from her, but you will get more information about our newsletter and on our LinkedIn page through this new webinar in December. So Cosmin, thank you again for your great presentation, very insightful. I think it's very useful. And yeah, we are looking forward to your new research soon in
Thank
the
you. Thank
tube webinar.
you for this opportunity, and feel free to reach, all the participants, feel free to reach out if you have any further question. I'll be happy to answer.
Thank you very much. Have a great day and talk to you soon.
Thank
Bye-bye.
you. Bye-bye. Bye.